cancer can occur in most parts of the body, but cancer is generally divided into four main groups: 90% epithelial cancer, approximately 10% lymphoma cancer, fat cancer. Nearly 1% is endothelial cancer, blood cancer, and especially brain cancer. What is the difference between traditional medicine and precision medicine in cancer treatment? Precision medicine is an effective and specific method of targeting cancer. In precision medicine has three main branches: genetics, immunotherapy, targeted therapies. For each major branch, we have different approaches with varying levels of access from DNA to patients. So, what is the main process of precision cancer medicine? Patients first need a molecular diagnostic test to identify cancer, an abnormal origin when compared to a previously known cancer, sequence or genome, and finally suitable targeting therapies are suggested. Although there are three main directions in precision medicine, precision immunotherapy is the one that is most applicable and is considered safe because it focuses on stimulating the immune system of a patient to self-attack cancer cells. First, they alert the immune system that cancer-specific antigens or antigens that are abundantly present on cancer cells are foreign. Second, immunotherapy stimulates the immune cells to attack cells that have these antigens on their surface. Immune cells called dendritic cells start to eat the invaders and their non-self antigens. This process causes the dendritic cells to transform into antigen-presenting cells, APCs. These APCs expose the invader cells to the primary immune cells of the immune system, the B and T lymphocytes. These cells can recognize the invading cells and work to destroy them. B cells work rapidly to produce antibodies, and T cells are activated, then multiply into an army equipped with necessary weapons to defeat the invader. In the world, scientists have applied different types of immunotherapy. There are types of interferon, interleukin, or colony-stimulating factors, cytokines, but they are not recommended because these therapies are not effective. In addition, they have the side effect of attacking normal cells along with cancer cells. To overcome these disadvantages, precision immunotherapy has been introduced with therapies such as immune checkpoint inhibitors, chimeric antigen receptors, CAR, T-cell immunotherapy, and vaccines. First, we will learn about immune checkpoint inhibitors. A patient's cancer cells can express molecules that activate PD-1 or CTLA-4 inhibitory receptors on their T cells or other cells in the immune system. When these receptors are activated on the T cells, they are prevented from attacking the cancer cells and evade the immune response. Checkpoint inhibitor drugs that block PD-1, PD-L1, or CTLA-4 work to release the brakes, allowing the cancer cells to be detected and attacked by T cells. The second is the chimeric antigen receptor CAR T-cell immunotherapy. T cells are collected and reprogrammed in the laboratory to recognize and attack a patient's cancer cells. Once the T cells multiply and reach a certain number in the laboratory, usually hundreds of millions to billions, they are reinfused into the patient. The infused T cells then circulate throughout the body, attacking the patient's cancer cells. The key step is to genetically modify a patient's T cells to express a CAR that is designed to target an antigen protein described on the cell surface. As a result, the reprogrammed T cells, or CAR T cells, make protein that bind and attach to antigens on cancer cells to help destroy the cancer cells. CAR T cells appear promising for the treatment of B-cell acute lymphoblastic leukemia and other hematologic malignancies. In this, therapy are divided into two main categories: CTLA-19 CAR T-cell therapy and BCMA CAR T-cell therapy. The first therapy teaches T cells to recognize and attack myeloma cells. CTLA-19 is designed to attack myeloma stem cells, a cell type that can give rise to many more myeloma cells. A pilot study reported that this approach was safe and feasible. The second therapy teaches T cells to recognize myeloma cells through a protein called B-cell maturation antigen, BCMA. This approach was reported to have promise in treating myeloma in patients who had already failed other therapies. The last major direction is the vaccine. A vaccine is designed to stimulate a response by the immune system against specific targets or antigens. When cells in the immune system perceive the antigen, which is part of the vaccine, they multiply to fight it off. Vaccines can be either preventive or therapeutic. The cervical cancer vaccine Gardasil is a preventive vaccine. It prevents cervical cancer in women by providing protection against several strains of the human papillomavirus, including those responsible for causing many cases of cervical cancer. Therapeutic vaccines, by contrast, are designed to stimulate the cells of the immune system to kill cancer cells, stop a tumor from growing, or stop a tumor from coming back. Provenge Sibelisol T was the first dendritic cell vaccine. Its intended use is for the treatment of metastatic hormone-refractory prostate cancer. Provenge is custom-made for each patient. First, a patient's immune cells are collected and exposed to a protein that is found in most prostate cancers linked to an immune-stimulating substance. Next, the patient's own cells are returned to the patient to stimulate an immune response against